So next topic is DNA replication. DNA replication is a very important process because that is involved in the formation of DNA, new DNA molecules from the parent DNA molecules. And this is the beauty of DNA replication, that in 1 trillion cells of human being, each of these cells is genetically identical to the zygote from which they are formed. For this to happen, the genome must be copied quickly, the genome must be copied accurately or precisely, and that only can happen through the process of DNA replication. Replication of DNA, the DNA replicates by making strand that is complementary to each original strand. What is the objective of these slides? To summarize the role of enzymes involved in the replication of DNA and explain how leading and lagging strands are synthesized differently. Template strand, a molecule of DNA that is a pattern for the synthesis of new strand, that is called template strand. And we will also discuss about the semi-conservative replication, DNA polymerase, and Okazaki fragments. Comparing DNA replication in eukaryotes and prokaryotes, how the DNA replication is different in eukaryotes and prokaryotes. Eukaryotic DNA unwinds in multiple areas as DNA is replicated, while in case of prokaryotes, the circular DNA strand is opened at one origin of replication only. So this is the difference between eukaryotic DNA and prokaryotic DNA replication. There are different models of DNA replication. Number one is conservative replication, second is dispersive replication, and the third is semi-conservative replication. So the conservative replication would leave intact the original DNA molecules and generate a completely new DNA molecules. And dispersive replication would produce two DNA strands, DNA molecules with sections of both old and new DNA interspersed along each other, along each strand. Semi-conservative, however, that is the most accepted kind of model of DNA replication. The semi-conservative replication would produce DNA molecules with both old and new DNA strands. But each molecule would be composed of one old strand. Here you can see one old strand and one new strand. This is again the elaboration of this semi-conservative replication. Here the parent strands unwind from each other, separate from each other, and they act as templates for the production of new DNA strands or daughter DNA strands. So in the new DNA strand, one new DNA molecule, one strand is the old or parent strand, and the other strand is the daughter strand. So it starts with the joining of the RNA primers with the unwinded DNA molecules. These primers are made up of RNA, and DNA polymerase removes the RNA primers and fills in the place with the DNA nucleotides. DNA ligase links the two sections or fill the gaps or nicks between the two sections. There are different stages of replication process. Number one is initiation, number two is elongation, and number three is termination. In initiation, the DNA double helical structure begins to unzip itself through the through the help of different enzymes which are called helicases. DNA is tightly bound, stable structure for most of cell's life. However, in replication, that is unzipped. DNA unwinds at special points along the strands, and these special points are called replication forks. Enzymes called helicases are involved for the unzipping of the DNA molecules from each other. Elongation, assembly of DNA, new DNA strands begins by the help of DNA polymerases, and newly replicated DNA can be found in short segments called Okazaki fragments ranging from 1 to 2000 nucleotides in length on the lagging strand. Replication in 5' and to 3' direction on one strand, that is called leading strand, and it occurs in 3' and to 5' direction on the other strand. The enzyme DNA primase begins this process of formation of a new strand. Leading strand, that is the strand replicating in 5' and to 3' direction. Lagging strand replicates in 3' and to 5' direction. And Okazaki fragments are joined together by the enzyme called ligase enzyme, DNA ligase. Here we can see the diagram of this replication fork. The DNA strand, the leading strand unwinds with the help of this helicase. The double strand of DNA unwinds with the help of helicase enzyme, and leading strand is formed where there is continuous formation of the new strand. However, on the lagging strand, Okazaki fragments are formed. And that is discontinuous formation of the new strand on the lagging strand. And on the lagging strand, the formation is from 3' and 5', and on the leading strand, the formation of new DNA molecule is from 5' and to 3'. Because one strand is synthesized continuously and the other discontinuously, DNA replication is said to be a semi-continuous process as well as the semi-conservative process. Termination, how termination takes place, the stage when the new DNA molecules reforms in helical structures or double helices, daughter DNA strands rewind, forming their stable helical structure. Each of the new daughter DNA molecule is slightly shorter than the parent DNA molecule. Chromosomes lose about 100 base pairs with each replication, and that is mainly at the telomeres, at the caps of the chromosomes. What are telomeres? Like the hard ends of the shoelaces, telomeres are the protective caps of the DNA molecules, and that occur towards the end of the chromosomes. Eukaryotic cells, special regions called telomeres, which have the base sequence of TT, AT, GGG, are attached to the ends of each chromosome. These sequences have no role in the development, and thus the chromosomes can lose them with each replication and not lose any important genetic information. However, it is speculated that somehow from telomeres we can have an idea about the aging of the cells. As to R is to human, and also R is to the DNA replication as well. So there are some mismatches in the DNA replication. There are some errors in the DNA replication or mistakes. Though we would like to believe that DNA replication is a very precise step-by-step process, This is usually not the case. Just as we make mistakes, DNA replication process can also make the mistakes, that is in the form of wrong bases may be inserted in the new DNA molecule. Nucleotide bases may be damaged by radiation, for example. When this happens, mutations or other serious problems can occur in the DNA molecule. So, for this proofreading and correction of the DNA molecule or of these mutation and mistakes in the DNA molecule is needed. To prevent errors from occurring, the enzymes DNA polymerase is able to check to see whether bases are actually bonding together by hydrogen bonding. No hydrogen bonding means there is a mismatch, base pair mismatch. The incorrect base pair is replaced with the correct one in the proofreading. The DNA replication involves dozens of different enzymes and other proteins which work together as a replication machine to get the job done correctly, precisely, and virtually error-free. So that was all about the DNA replication and proofreading.